Hi, good morning, good evening, good night, wherever you are. My name is Jeff, and I'm one of the business developers for Rousselot Biomedical. And I want to talk to you today about unlocking the next step in gelatin-based biomaterials. Um, I work for Rousselot, and we've been making gelatin and collagen-based products for 130 years. Not sure if you know us in the biomedical world, but there's a high chance you've already eaten or consumed our products, whether it's in hard capsules, soft capsules, food products, nutritional supplements. They're quite ubiquitous. Now the question is, why does a company like Rousselot move into the clinical world? And why, before we go into details of clinical translation, um, well, why? Because we want to make a difference. We want to advance medical science. We want to help you create solutions that sustain life. But put it bluntly, we make gelatins. So how do we think we can achieve this? How can we contribute to advancing medical science? Well, the answer is simple: by removing a hurdle or removing a barrier. And I will use GelMA or gelatin methacryloyl. I think that's all well known by you all, and I will use that as an example. Now, GelMA isn't new. It's been around for I think the year 2000 when it was invented at Ghent University, and since its inception, the vast list of applications has sprung up, from sustained drug delivery, microneedles, adhesive, cell encapsulation, tissue regeneration, bone reconstruction, 3D printing. The list of scientific publications and patent applications has grown every year. Yet, to our knowledge, and by all means, if we've missed it, let us know. But to our knowledge, none of these GelMA applications has made it to a patient yet. And then the question is, what's the holdup? Were all these scientists and developers wrong? Is that some kind of a major drawback they didn't know of? Well, the answer is no. The answer seems to be quite simple. There wasn't enough of the product available in terms of quantity, and what was available was not always consistent in terms of quality, and as such is not usable for clinical trials. There are plenty of companies today offering a few grams, tens of grams of research grade unpurified GelMA in vials or syringes, which might be useful for academic research, but it is meaningless if you need a few kilograms of high purity GelMA every year for your medical applications. And that is where we believe we can make a difference. We can, by unlocking the next level in the development of gelatin-based biomaterials, which is the clinical trials, by allowing developers to bring their gelatin-based products or gelatin-containing products, devices or therapies to actual human beings. And back to where we started. This is where we Rousselot come in. We had a customer that was facing this exact challenge. They had a great idea for a medical device, but if their clinical trial is successful, they will need a steady supply of a few kilograms of GMP GelMA every year. And they couldn't find anyone who could supply this. And to be fair, when we first met him, which of course was the December 2019, we didn't know if we could do it either. But we knew how to modify gelatin, and we already had to process the purified gelatin. We also have a large direct access. We also have direct access to large amounts of suitable, high-quality starting materials. So we decided to take the leap. And I'm saying in a few weeks' time, my R&D colleagues developed and tested the GelMA production and purification process that could later be GMP certified, which we're now, in the final stages of doing. But it was also scalable, and to me, that scalable part is equally, important as the GMP part, because obviously, it needs to be suitable for use in medical devices and drug products, hence GMP and purity. But scalability is what makes the development process so much easier, since what we supply now to customers on a 10 gram or 100 gram scale as research material is equivalent to the final GMP material if it's 1 or 10 or 100 kilogram scale. Now, to illustrate the importance of scalability, I'll go back to that first GelMA customer. Today, we are supplying them with a few hundreds of grams of various GelMA types at a time, which they use in product development and preclinical animal trials. But they have the intention of going to patients by early 22. And this is where the scalability matters. Because our process is scalable, what we produce on our lab scale installation will be equivalent to the final GMP GelMA. So for these customers of ours, this equivalence means they can easily move from their current preclinical testing phase with our research grade material to their first in-human trials with the GMP material early next year. There's no need for them to revalidate their design or to reevaluate the GelMA once it's GMP. It is like for like the same product, full stop. And while they are fine-tuning their product and doing their preclinical work, we are building a larger production facility where we're producing kilogram batches by what we are now, and we'll be fully up and running by the end of the year to make GelMA under the appropriately certified GMP conditions. But there's GelMA is one example of how Rousselot can co-develop, but we can actually go one step further, and we've launched this I think in May, and it's very proud to also talk about this. It's a same unique value proposition. It's high purity, low in toxin, low impurity, very consistent product, which is available in commercial quantities and produced with a suitable GMP process. And like GelMA, we developed this product in collaboration with a company, looking for a specific modified gelatin. But unlike GelMA, where we could use an existing modification, this new modified gelatin required us, to go one step further back and revise the synthesis. What our customer wanted us to make was gelatin tyramine, which is what we set out to make. However, when we first made this according to our customer's process, we failed miserably. I won't go into the nitty-gritty scientific details right now, but the process described in literature to make gelatin tyramine uses EDC and HS, and just a record, EDC and HS does not scale well. It yields inferior products because of side reactions, cross-linking, and so on. We simply destroyed and cross-linked the gelatin in the process. It just so happens we have some clever people in R&D, wasn't me for the record, and they came up with a rather elegant alternative that gives a gelatin with the same, I would say, a phenolic flavor or same phenolic functionality like gelatin tyramine. And if you would look from far, it looks the same, but which we can produce and purify at the right scale and the right quality. So in short, we ended up revising the synthesis reaction to create a product that has the functionality our customer needs, but is better, I think, in almost every other way in terms of process complexity, purity, safety, toxicology, and so on. What my colleagues came up with was gelatin desaminotyrosine, or GelDot for short. It is a modified gelatin, which is functionally equivalent to gelatin tyramine, which what our customer needed. It is similar in juice to GelMA. It can also be UV cross-linked with the correct potentiator. But additionally, it can be enzymatically cross-linked, and because the phenol groups are less specific in their reactions, it is also more adhesive than GelMA. For example, it can be used for direct on-cell cross-linking. And another similarity to our GelMA is currently being used in preclinical animal trials and also hoping to go to human trials in the next few years. Now, I talked a lot about the why we're in this business and how we make a difference. What do we actually offer? 3 existing product ranges: purified GMP gelatins, gelatin grade or hydrolyzed; purified GelMA, will be GMP by the end of this year; and our purified GelDot, which we can turn into a GMP quality if the need arises. Now, all our products can be, and they are regularly customized for our customers based on the application. So we can change the molecular weight, we can change the humidification depending on whether you want a higher gel strength or lower viscosity, either way. And I hope I've given enough proof that we are able to co-develop. So if you have your own idea for a clinical application using a GelMA or a gel dot or any other modification, if you have some questions on the GelMA or anything else, a prototype or an idea, and you think you might be able to use GelMA, if you want to collaborate in any way or shape or form, please talk to us. I'm always willing to listen, and you can reach me at the address shown here. Now, I wish you all a great morning, a great evening, or rest of the day, whatever you are, and I hope to speak to some of you very soon. Take care.